Cells. They make up all life. From the simple bacteria, too small for the human eye to see, to the massive blue whale traveling through the sea, cells are everywhere. Cells, of course, must protect their contents from the outside while allowing and keeping important nutrients in. The main part of the cell membrane, which is what keeps unwanted substances out, is the phospholipid bilayer. It is made of two layers, phospholipids with polar heads on the outside and nonpolar tails in the middle. The cell membrane also contains many proteins, consisting of two types: peripheral proteins that only touch one side of the bilayer, and integral proteins that touch both sides. Some proteins are glycoproteins, meaning the protein has carbohydrate chains attached to it. They are generally involved in cell-to-cell interactions. There are also glycolipids, which are carbohydrate chains with fatty acid chains. They are important in cell recognition. While the cell membrane does generally succeed in keeping out most hostile substances, it also allows many substances to enter in and exit out. Some will do so through diffusion. Diffusion is a process through which molecules disperse, tending to move towards equal concentration, or an isotonic situation. Some molecules, like oxygen and carbon dioxide, will go through the phospholipid bilayer. As we can see here, we have a higher concentration of a substance inside of the cell, a hypertonic scenario. As a consequence, some molecules will travel through the phospholipid bilayer, creating an isotonic situation. Note that if the concentration of a substance is less inside of a cell, the situation is considered hypotonic. Sometimes, however, some substances, like glucose amino acids, are too large or too polar to simply pass through the cell wall. In these cases, the substance will pass through using a channel protein. This is called facilitated diffusion. As we can see, a substance is traveling through the channel protein from higher to lower concentration. A well-known case of facilitated diffusion is with water, in which a solvent, generally water, moves through an aquaporin to allow for an equal concentration of a solute inside and outside of a cell. This plays a key role in plant cells, which depend on osmotic pressure created due to water in a cell for structure. Sometimes a cell will try to have a more extreme concentration, or lack thereof, of a substance. In these scenarios, active transport is necessary. This is where the sodium-potassium pump comes in, and Jason Gahn will explain.
Hey, this is ATP. Here I am inside of the cell. I'm going to help these three here, sodiums, get outside of the cell through this sodium-potassium pump. And I'm going to let these two potassiums in. So as sodium molecules, they are unable to get through this door because they have no hands and they can't push this open. So what I'm going to do is I'm going to press this here and I'm going to activate the pump, and then these three sodiums are going to walk out. And once these three sodiums leave, these two potassiums are going to come in. So as you can see, they came in. And now that they came in, the pump eventually transforms back to normal.
There are other ways for the cell membrane to allow molecules in and out. These other methods are endo and exocytosis, which use vesicles, circular rings of phospholipids, to draw substances in and out. Here we see a wild bacteria, unaware of the treacherous territory it has walked into. As it nears the cell membrane of the cell, its final moments are at hand, as the cell has detected its prey. And with a quick grab, it is absorbed. The cell membrane then secures its prey by creating a pocket that pinches off, also called a vesicle. This process is known as phagocytosis, and is a means of feeding for single-celled organisms. Phagocytosis exists in the human body as a defense mechanism against infection and foreign substances. Yet another instance of particles falling victim to the cell membrane can be seen in this next clip. As the molecules outside the cell membrane move around outside the cell, the membrane reaches out and surrounds the helpless pair of molecules. The molecules are trapped inside a vesicle and cannot get out. The cell then brings its prey in for processing. This process is another form of endocytosis, where a cell takes in a random assortment of particles. If a cell wishes to eject substances, it will do the reverse. This is called exocytosis. Exocytosis is demonstrated in this clip, as the world-famous vlogger Rotomolecule attempts an internet challenge.
What up, guys? It's Rotomolecule here. I'm chilling at the cell membrane right now. I don't know if you guys can see it, but so I'm here with this cell membrane jacket, and what I'm gonna try to do is the exocytosis challenge, where I leave the cell with the cell membrane jacket. Let's see how it goes. Hello, gentlemen.
Okay, you can leave that.
What?
You may be asking, why couldn't Rotomolecule
What up, guys? It's Rotomolecule here.
leave with the cell membrane jacket, also known as a vesicle? This is because the substance must have the vesicle removed by the cell membrane, so it can add the phospholipids of the vesicle to the cell membrane. This has been a lesson in cellular biology. Thank you, and have a great bio period.